By now you should be professionals to pick up these planaria and move them over. In this video, I will walk you through the behavior experiment you will conduct during next lab's session. For this, we will be measuring locomotion. That means counting how many times the worm crosses a line at specific time intervals. First part of the experiment will be conducted with a control substance. In our case, artificial pond water, APW, or sometimes labeled here for you guys as planaria water. So everyone will have one of these tubes in front of them. So the first thing that we want to do is we want to get our measurement dish ready. So you grab one of these big dishes and transfer a total of 20 milliliters of artificial pond water into this Petri dish. For that, we will be using the 10 milliliter pipette together with the pipette pump. So we will pull up 10 milliliters into the pipette, transfer that over here one time, and we do the same thing the second time, and you will see very quickly after we transferred the 20 milliliter of liquid into the dish, we will have nice coverage of the whole dish with liquid, giving the planaria enough room to move. So now that we have the liquid ready, we move over and grab our planaria. For this experiment, we actually will be using the brown planaria that you can see here. So what you will do is you will grab one of these little transfer pipettes, and by now you should be professionals to pick up these planaria and move them over to the bigger dish. At this point, we can actually start our stopwatch and give the planarium 2 minutes in order to acclimate. While they're acclimating, we grab our graph paper or 1 centimeter by 1 centimeter paper and place it onto the microscope. Will look something like this. Now that we have that on there, we can center our planarium right on top of that, which gets us ready for measurement. While the planaria take their 2 minutes to acclimate, we will take the time in order to get our Excel sheet ready. So you will open up Excel, and we need a total of 2 columns for this experiment. On one side, the incubation time in minutes, and we measure a total of 5 minutes, and then on the other hand we start with our artificial pond water. So let's have a look. 1.5 minutes. Now once we have our planaria ready, we also want to make sure that our microscope is adjusted right. So in order to do that, you will grab the big knob in the back and move it up and down until you get a sharp image, like I do right here. Once I've done that, I want to make sure that the small knob that is operatable on the left and the right is all the way counterclockwise so that we have the maximum view to track our planaria. Almost ready. By now, our planarium has made its way to the corner, which means we have to actually get them out and place them back into the middle. Now we're going to find them. So by now we have all decided in the lab what we define as the head crossing one of the lines. And that's what we will exactly use in order to track that.
So as I'm by myself, we're going to see how that works. You certainly can use your teammate to manage the Excel sheet while you're counting the movements of the Planarian.
So we finally hit the 2-minute mark and the Planarian should be acclimated.
So what I would do now is to move the planarian in the center of the dish again and start observing the planarian after I started my timer. Again, the teammate manages the Excel sheet while you are counting.
So let's have a look at the microscope and start our timer. Let's go. We will count every time the planaria moves. We can use the paper. I have 1, 2, 3, 4. 5. I will use the paper in order for the planaria to stay in focus. I have a total of 5 times the head that passed over. Only swimming here on the top, so I'm gonna count 6. Let's see if it's gonna start swimming again.
At this point, you see me move the paper and the dish, which also move the planarian across some grid lines. For you, I recommend stopping the timer if the planarian stops moving or starts floating. This way, you can relocate the planarian to encourage movement and start the time back again.
7. Okay, that was 7. I keep counting. 8, 9, 10, 11, here should be 7, 13, 14, 15, 16, 17, 18, 19. We keep track. 20. So you will see at this point our planaria has reached the corner, so we will stop our timer. That will be the same for you. For us, when we now look at the focus here, our planaria made its way all the way to the edge. So we will not measure while it is at the edge. We will stop the timer, make sure that we have our numbers here in check. So let's have a look very quick at the Excel sheet. My classmate or teammate will have logged that we had 7 and then a total of 15 in here. And while we stop the timer, we move our planaria back into the center. In order to continue the experiment. But it will be done a total of 5 minutes, and let's say 34.
All right, now that we have our sample data collected, we want to analyze and visualize that. So the first thing that we have to do is write down what we collect, which is the controls. We collected the cumulative crosses here. So the cumulative means we counted them up. But what we want to actually plot out is the APW average crosses per minute. So how do we calculate the average crosses? For the first value, it's very simple because it's the same per minute. And then for the second, we subtract the previous cumulative crosses from the one that we currently measured. So we now have 8, 6, 7, and 6 crosses per minute. And you see the formula was pulled into this. Now that we can have the average number of crosses, we can plot that out to just have an overview. So we go insert, scatter plot, and I like a smooth line. So we select our data, which would be APW, which is the control. That's we're going to plot out control. For our X value, we're going to plot out the time, and for our Y, it would be average number of crosses. So the first thing that you can see now in the plot here, and we're going to add the axis title so that everybody can really see it, is here time in minutes, and this is the average number process per minute is that there's a slight downward trend here. Not too bad, but there's also natural variation. Obviously, it goes a little bit faster, then slower, then faster and slower. Now, there's one wonderful thing that we can do is, let's say we measured for the caffeine, the cumulative process. We can actually normalize our results. So let's pretend we collected data. So let's say 15, we collected 30, we collected 44, we collected 56, and in the last one, we collected, I don't know, 69. All right, again, we're having our average crosses, this time again for caffeine. And we do the same trick, same. And this one minus the previous cumulative crosses. We pull that in and we see, okay, the average crosses vary again a little bit. We could plot that on here just by adding that data. Oops. Select the data. We add our next value. Again, the times are the same. This one has to be these cumulative crosses, and it's what we caffeine. So now we can basically plot this out, and we can see there is a little bit of variation for the caffeine too. So this is the overall idea. We could even do a t-test between the not of cumulative, the average crosses versus the second average crosses. We have a one, let's say if they're just different, two-tailed t-test, and they're not paired in our case. So we're going to go with 2. So there is a significant difference between the two, and that makes sense. We see this. But we can also plot out the average, which is average crosses here versus average crosses here for the whole time, because it doesn't really matter if we measured for 5 minutes or not. This gives us just an idea if they slowed down or not. Or one thing that you will see actually in science done very often, if we were to measure the very same planarian, so we measure the planarian one time in water and then in caffeine, we can actually normalize. Like, let's say after 5 minutes of under the microscope, they really are slowing down just naturally. So what we can do is now normalized caffeine motility. Very important. Normalized values don't have values, they're ratios, because we can say how much faster is the caffeine compared to our average crosses. So we divide to each other. So it's about 2 times faster. And we normalize that. And what you can see now here is the normalized value is kind of interesting because it fluctuates also over time. If we have like a slowing down over time, with the normalization, we can get rid of that. Because if both planarians are slowing down over time, then the values here in APW will get smaller, as well as the values in the caffeine. So let's plot that out very quick too, so that you can have an understanding on what that looks like. We select our data. Same thing. Now we're going to call it normalized caffeine motility. X values still the same. Y values would be the normalized value. And what you see here with the normalized values, you have now like a pretty much like robust variation between the two. Let's pretend the crosses slow down. So we're going 23 and we're going 25. So they're like, 24. So they're really slowing down. And we see the very same thing also here for the cumulative ones. So what do you see here now? If we look at the average cluster, they go 7, 8, 6, 2, 1. However, if they go the very same steps here in our caffeine 2, even though there's a slowing down, we can now, with the normalization, take care of that. So we can say that even though the planarians both slow down, because we observe the same effect in our controls as well as in our samples, that's normal and fine. All right. So that's how we plot out our data. You can either report it here on the left side with, obviously, we're going to add the legend, or you can report the normalized values. That's up to you. You can also report our averages with a bar plot.
All right, that's as far as the experiment for lab number 1 goes. I hope you enjoyed the video so far, and I see you when you actually get to do this.